example as your friend is being faced with a danger. Let's say a dog is about to attack your friend. You know, the first thing the dog will do is to start barking, you understand? So what your friend will do is going to pick up the barking sound of this dog through his auditory pathways, you understand? So first, let's draw what actually happens in the normal human body. Let's say this is the cortex. It's an example. Your cortex is not rectangular in shape, by the way. So as your friend picks up this barking sound through the auditory pathways, it gets to his temporal lobe. Let's use T, his temporal lobe. So from the temporal lobe is going to relate the information to the limbic system, guys. To the limbic system. Okay, now then, as this information gets to the limbic system, your friend is now going to turn and see what is actually barking. So when he turns, he's going to see the dog, and then he's going to pick up this sensation through his auditory, his visual pathways. From the visual pathways, information is going to be related to the occipital lobe. When the information gets to the occipital lobe, what the occipital lobe does is that it's also relating information further to the limbic system. So now the limbic system is now bombarded with information from the visual pathways and also from the auditory pathways. You understand? So, but we also have information coming from the cerebral cortex, maybe due to previous experiences. You understand? Maybe your friend has seen a movie where he must have seen a dog attacking an individual. So the cerebral cortex is going to relate the same information to the limbic system. So now we are getting information from three places: the auditory pathways, the visual pathways, and also the cerebral cortex relating to the previous experiences. That's memory, guys. Okay, what happens is that the limbic system, on the other hand, is now going to relate this same information that is being bombarded to the hypothalamus, because the hypothalamus is the control center of the autonomic nervous system. But in the hypothalamus, we have the anteromedial nucleus, which relates basically with parasympathetic, and also we have the posteromedial nucleus, which relates basically with the sympathetic. But under normal condition, these two sides of the hypothalamus, they are in equilibrium. So the information from the limbic system is going to come and disturb this equilibrium that exists. You understand? So what actually happens is that the information from the limbic system is going to stimulate the posterolateral nucleus of the hypothalamus, while simultaneously it's going to inhibit the anteromedial nucleus of the hypothalamus, guys. So when that happens, there's going to be a shift in the normal equilibrium. For example, favoring the sympathetic aspect of the autonomic nervous system. You understand? So what actually happens is that when the posterolateral nucleus of the hypothalamus is overstimulated, the information gets down from the CNS through what is called a polyneuronal descending pathways. So now this polyneuronal descending pathways, they are network of neurons, guys, that descend from the posterolateral nucleus of the hypothalamus. What they do is that they transcend to the spinal cord, you understand? And in the spinal cord, they go to the segment of the spinal cord that relate to the sympathetic nervous system. Where are we talking about here, guys? We are talking about from T1 to L2. Sometimes it could be L3, depending on anatomical variation, guys. So now, what actually happens there? From this point in time, the information which is being which comes from which comes from the posterolateral nucleus through the polyneuronal descending pathways gets to this segment of the spinal cord, T1, L2, or L3. So now this information is going to go out from the spinal cord, that's from the CNS, through what we call the sympathetic outflow. You understand? Now this sympathetic outflow, we're talking about this preganglionic sympathetic fibers, guys. You understand? And this preganglionic sympathetic fibers, or we can call them the sympathetic outflow, sympathetic outflow, they are actually they are axons. They are axons of the the neurons which are found in the lateral horn of the spinal cord. You understand? Their cell bodies lies in the lateral horn of the spinal cord. So these axons, they now travel to the what? To the sympathetic ganglion, guys. So from the information coming from the polyneuronal descending pathway hit the cell body, understand, in the lateral horn. You understand? So this information is now related through what the preganglionic fibers. You understand? So when it gets to the sympathetic ganglion, you understand? From there, the preganglionic fibers, they release acetylcholine, and they stimulate the nicotinic receptor, which is found in the sympathetic ganglion. When they stimulate the nicotinic receptors found in the sympathetic ganglion, the information is now further related to the effector side through the post-ganglionic sympathetic fibers, guys. You understand? When this information is related to the post-ganglionic sympathetic fibers, now at the terminal point of the post-ganglionic sympathetic fibers, that is where the main action takes place, guys. That's where the main thing lies, because it is at the terminal point of the sympathetic fibers, these post-ganglionic sympathetic fibers, that we have the release of the neurotransmitters. And when we're talking about the sympathetic nervous system, which neurotransmitter are we talking about here? We're talking about the norepinephrine. But mind you, these preganglionic fibers, they can also go to the adrenal gland. You understand? When they go to the adrenal gland, they stimulate the cells which are found in the adrenal gland. And when they stimulate the cells which are found in the adrenal gland, what does the adrenal gland do? The adrenal gland now release epinephrine into the systemic circulation. You understand? In which the epinephrine finds its way to this particular adrenergic receptors, and it stimulates them, and to bring about the corresponding responses. You understand? So what actually happened is that at the nerve terminal, that is where the synthesis of the norepinephrine takes place, because norepinephrine is being synthesized in the nerve terminals, guys. So what actually happened? How does norepinephrine get synthesized, guys? We know that norepinephrine is synthesized from an amino acid called tyrosine, guys. Tyrosine. We have tyrosine. Eventually, in the nerve terminal of this particular sympathetic fibers, we have special tyrosine pumps. You understand? They are specialized in taking tyrosine from the circulation system is from the blood. So what happens is that when tyrosine gets into the exoplasm, guys, you understand? Or when it gets into the cytoplasm of the nerve terminals, it meets an enzyme called tyrosine hydroxylase. Now, this tyrosine hydroxylase convert tyrosine into dopa. You understand? So now, this particular dopa now gets converted to dopamine by dopa decarboxylase, guys. You understand? So now, in the same exoplasm, there are what we call spatial pump in the vesicles, which are found in the terminal part of the neurons. So those vesicles, they have spatial transport pumps that transport dopamine into the vesicles. So inside the vesicles, which are found in the terminal part of the neurons, dopamine gets converted into norepinephrine by an enzyme known as called dopamine beta hydroxylase. Dopamine beta hydroxylase. This particular enzyme brings about the addition of a hydroxy group on the beta carbon found on the structure of the dopamine, guys. So it's after the conversion of this particular dopamine into norepinephrine. Now you agree with me that more dopamine is being transported into the vesicles. You understand? And as a result of that, you understand, there will be a build-up of dopamine inside the vesicles. So how does the body take care of this particular situation? Because if there is consistent build-up of norepinephrine inside the vesicles, there is going to be a build-up in osmotic pressure. And once there is a build-up in osmotic pressure, guys, After a particular certain period of time, there is that tendency that this vesicle can what? It can burst. So inside these vesicles, there are special molecules which are known as what? Chromogranin, guys. So now this particular chromogranin, what it does is that it binds the numerous molecules of norepinephrine together so as to lower the osmotic pressure inside the vesicles. Inside